The overall goal of this step-by-step protocol is to quantify the abundance of cortex fragments, or reducing sugars, during bacterial spore germination.
This method will detail the steps required to detect the presence of cortex fragments or reducing sugars during spore germination.
The main advantage of this technique is most laboratories have the reagents and equipment necessary to successfully complete the protocol.
Begin this procedure by placing a tube of Clostridium difficile spores in a heating block and incubating at 65 degrees Celsius for 30 minutes. After the incubation, place the tube on ice.
The germination solution is specific to the species of bacterial spore being studied. For assaying C. difficile spore germination, we use a solution of 10 millimolar tris at pH 7.5 with 150 millimolar sodium chloride, 100 millimolar glycine, and 10 millimolar taurocholic acid and deionized water.
Prepare 1 milliliter of germination solution per sample, plus an additional 1 milliliter. Before beginning the assay, transfer 1 milliliter of the germination solution to a microcentrifuge tube and set it aside. This will serve as a blank for cortex fragment detection. Next, to the germination solution, add spores to an OD600 of about 3.0 and briefly vortex. This amount of spores works well for monitoring cortex fragment release during C. difficile spore germination. Immediately after adding the spores, remove a 1.2 milliliter sample to a new microcentrifuge tube and put it into the centrifuge. Then, centrifuge at room temperature for 1 minute at 14,000 times G. This is the zero time point.